Hello friends, this is 6th part in the first year syllabus, Organic Chemistry, some basic principles and techniques. Till now we have studied some purification techniques and the same is continued in this session also. You have learnt about chromatography in the last session. Under chromatography there were two types, one was adsorption chromatography, the other was partition chromatography. Under adsorption chromatography were two types again. The first was column chromatography and the second was thin layer chromatography. You have studied only column chromatography. So today we shall begin with the second one, that is thin layer chromatography. It is thin layer chromatography. Thin layer chromatography is a type of adsorption chromatography. This means it works on the principle of adsorption. I have told you in my last session that different components, different compounds get adsorbed onto a stationary phase to different extents. Whichever gets adsorbed more travels less distance, and that compound which is less adsorbed, which is loosely adsorbed, it will travel more distance. This principle helps us in identifying the separated components in a column. In column, separate bands or layers were visible, due to which their elution, their separation was easier. How do we carry out this in thin layer chromatography? So, in thin layer chromatography, we again use the same stationary phases like silica gel or alumina. This time it is not packed in a column, but it is coated or it is pasted on a glass plate. Just as you have got slides in biology lab, similarly we have long rectangular glass plates, we call them as thin layer chromatography plates. Onto this plate we coat a layer of the stationary phase like silica gel or alumina gel in the form of slurry. What is slurry? Slurry is a very thin paste of either silica gel or alumina prepared in suitable solvent. In that slurry you dip a glass plate, remove it out, keep it for drying. Leave some space in any one corner of the plate for holding. So say you have got a glass plate, of this size, then from here up to here you coat the entire part with the stationary phase. What is our stationary phase? It is either silica gel or alumina. So a glass plate coated with the stationary phase is called thin layer chromatography plate, or it is otherwise called chromoplate. It is also called chromoplate. Now, just like in column chromatography where you had to separate 4 different compounds that were present in the form of a mixture, here also let us assume we have 4 different compounds which have got mixed. Now you want to separate them. How do you separate them? You prepare a solution of them in a suitable solvent. That suitable solvent should be same as the mobile phase. So we shall consider that as mobile phase. So a drop of that solution containing the compounds to be separated, a drop of that solution is now added onto this plate. How? From the lower end of the plate you mark a line. A line is marked at about 2 centimeters. At about 2 centimeters from here you mark a line. We call this line as, baseline. Okay. On this baseline the compounds mixture solution is put in the form of a spot. A drop of the solution containing the compounds to be separated is put or dropped or added on this baseline. How far this baseline should be from the lower end of the glass plate? At about 2 centimeters. Then this plate is left for drying. After drying, after the spot has properly dried, then this plate, TLC plate or chromo plate, is kept in a jar. It is then kept in a jar, a glass jar preferably. We call this glass jar as, developing chamber. We call this as developing chamber. What is developing chamber? It is nothing but a glass jar, an empty glass jar containing the mobile phase, containing a suitable solvent which we refer to as mobile phase. Okay. Now, there are some precautions that you need to take while keeping the glass plate. So, first I shall label this is our mobile phase, or you can also call it as solvent. Then what is on the glass plate? This is our stationary phase. This is our stationary phase. What is stationary phase? Either silica gel or alumina gel coated on the glass plate. While doing this work, make sure that while you insert the glass plate inside the developing chamber, the solvent should not touch the baseline. The solvent should not touch the baseline. You should also make sure that the spot does not come in contact with the solvent. Why? If it happens so that the spot comes in contact with the mobile phase, then this spot contains your compounds to be separated. All the compounds will get redissolved in the same mobile phase. They will not get separated on the plate. So, for that reason, while dipping the glass plate inside the solvent, you have to make sure that this plate, even if it is touching the base of the developing chamber, the solvent should not touch the baseline. Okay, this is main criteria. If it is not possible, then you can have any support over the glass plate for holding it. Now, if this is done, the glass chamber or the developing chamber is closed and this apparatus is left undisturbed for some time. When it is left undisturbed for some time, what happens? Now, this spot which was initially containing the compounds to be separated, they were of different colors. Now, they will get separated in the form of bands or in the form of spots. So, as and when the solvent rises up, when it touches the spot, it moves upwards. As the solvent rises upwards on the plate, it carries with it the compounds present in the mixture to be separated. So, based on their speeds, based on the speeds of the components present in the mixture, they get separated to different heights. How? You see, you have got 4 compounds A, say only 3 compounds A, B and C, and if A has got a speed of 50, B 60 and C 70, then whichever compound has got more speed, it will travel larger distances. So, after some time you will notice that there is one spot here, there is one spot here. After some time you may see there is another spot here, then you might notice another spot somewhere here. So, you will have 3 different colored spots, and at the last the solvent will also rise maybe up to here. After solvent travels till here, it might no longer move upwards. So, we say this is last line, and this line is called solvent front. It is called solvent front. What is solvent front? It is the maximum distance travelled by the solvent or the mobile phase. Okay. What is baseline? It is the starting line from where compounds or components begin to separate. Now, once you are able to notice that different compounds have been separated, then you remove this plate outside and let it for drying again. This time make sure that you do not dry the plate in open sunlight, as some compounds may lose their color in sunlight or else they may vaporize also. For that reason, you dry them in protected light or indirect light. Now if the compounds are colored, they are clearly visible, no? So after the plate has been removed, outside, then this plate, this TLC plate, which had 4 different spots starting from the baseline, one is here itself, the other is here, the next is here, and the last one is somewhere here. So this was our solvent front line. Okay. So after you have removed the plate and dried properly, then you using a blade, you mark this area, mark all the colored areas, After marking them, you remove this part in one separate beaker. You extract this powder. It will have what? It will have your organic compound along with the stationary phase. What is that stationary phase? Either silica gel or alumina in it. So you take this component in one beaker, keep it aside. Again take a separate beaker, extract this part, means scratch away that part in the powdered form in another beaker. So what will you do? You will have different beakers, for different compounds along with the stationary phases. Once this is done, you take the same solvent that you have chosen for mobile phase and add over the compounds present in the beaker now. Upon adding the solvent, the compound will dissolve in the solvent. Stationary phase will not dissolve. So you decant the solution or filter the solution. You will have now colored solutions. These colored solutions will contain your pure organic compound but along with mobile phase. So how do you separate? It is the same principle that you have followed in column chromatography. Once the solution colored solution comes out through the column, you collect them in different beakers. All these beakers solutions are subjected to filtration if required. Otherwise, then they are subjected to distillation or evaporation. Here also same. Once you separate the stationary phases from the compounds by dissolving the scratched portion in a beaker, that filtered colored solution is either subjected to evaporation or else it is subjected to distillation based on the nature of organic liquid. This is simple. Okay, it is the same as column chromatography, but it is somewhat different from column chromatography. Which technique? Thin layer chromatography is somewhat different from column chromatography. In what ways? Look, in column chromatography you were able to separate compounds based on their colored nature, that is fixed. So unless you have any colored compound, you cannot go for column chromatography. Means you need to have colored compounds only for column chromatography. But in thin layer chromatography, even if your compounds are colorless, they can be separated. How? You may ask, sir, because we could see different colored patches here, therefore we understood the compounds have separated. What if our compounds are colorless? Then you need to make sure the distance traveled by the solvent, once it reaches this part, it does not travel further. That you can visualize, that you can see clearly. So after making sure that the solvent is not able to rise more, it is not rising upwards, then you understand that your compounds have got separated. But because the spots are not properly visible, do not panic. You take out the plate, you take out the TLC plate or chromo plate, let it for drying, and after drying you have got different methods by which you can identify where your separated organic compounds are. See, if the compounds are colored, they are clearly visible on the TLC plate. If they are colorless, then you have different methods in which you can identify them. One, you can expose the TLC plate to UV light. You can expose the TLC plate to UV light. Upon exposing the plate to UV light, ultraviolet radiations, then wherever spots are there, only they will fluoresce, only they will glow. Fluorescing means only they will glow. The rest of the stationary phase looks as it is. but wherever your organic compound is, only that part will glow. So then what will you do? You will mark it with a needle. That portion you can mark with a needle, and then you can again scratch it away in separate beakers. Understood? If not, if UV radiations facility is not available, then what can be done? Then the second option is by keeping them in iodine chamber, by keeping the glass plates in iodine chamber. What is meant by an iodine chamber? It is again nothing but same kind of developing chamber, same kind of a glass jar containing some iodine crystals in it. So once the plate has been removed from the developing chamber, it has been properly dried, then you keep that plate, the dried plate, in a jar, closed jar containing iodine crystals. What will happen? Wherever the spots are there, only those parts will become brown. By exposing the plate to iodine, only the part or only the spot where compounds are present, they will become brown. Again, what will you do? You will mark that brown portion with a needle, scratch it away in a beaker, you will dissolve that in a solvent, filter it, and then subject it to distillation. What if iodine chamber is also not available? UV light is not available, iodine chamber is not available, then you can use the method of spraying. Spraying proper adequate reagents over the plate so that wherever compounds are present, only they will glow or only they will be identified. You might have seen this technique that forensic teams and crime investigation officers, they will use this for identifying blood stains at murder sites, at the crime scenes. They will spray some reagents due to which wherever blood is there, only that part will glow in the dark. So similarly, if UV light is not available and iodine chambers are not available, then you make use of this spray for spraying on the TLC plate, due to which only those parts which are having your organic compounds, only those parts will glow. So you can mark them with needle and get them scratched and isolated. So in this way you can separate any organic compound even if they are colorless by making use of thin layer chromatography. For example, spraying method is used for separating amino acids also. You have got many amino acids, then they can be separated or their presence can be identified on a TLC plate by spraying with a reagent called ninhydrin, by a reagent called ninhydrin. So these are the methods by which even colorless compounds can be separated in thin layer chromatography. So in this way, thin layer chromatography is advantageous over column chromatography. However, this technique has got one disadvantage: large samples cannot be separated. Large samples of organic compounds, if they are present in the form of a mixture, then they cannot be separated by thin layer chromatography, because you need to carry out multiple thin layer chromatographies at a time in order to separate them, and it becomes tedious and time consuming process. Only small samples, if they are present, they can be separated by thin layer chromatography. How is it advantageous? It is advantageous because even colorless compounds can be separated by thin layer chromatography, which is not so possible in column chromatography. And also there is one more advantage of thin layer chromatography. What is it? See, using thin layer chromatography, one advantage is that you can separate colorless compounds. Thin layer chromatography is again used to check the purity of a compound. Listen properly, it is used to check the purity of a compound which has already been purified by some other method. Say you purified a compound by other conventional methods, then you want to make sure whether the purified compound is correct or not, whether the compound is 100% purified or not. So that percentage of purity can also be decided by thin layer chromatography. Now, based on thin layer chromatography, you can also identify that the separated compounds have got what speed. The relative adsorption of different components present in a mixture, they are expressed by a term called RF value. RF value is nothing but retardation factor. It is nothing but retardation factor. Retardation factor is given by a formula A by B. Let us see now what A by B is. So say you had this glass plate, you had this TLC plate, this line was baseline, and till here say solvent travelled. So based on that, if this is solvent front and this is baseline, initially you had the spot on the baseline, isn't it? So from here, say one compound travelled a distance till here, okay, then another compound travelled a distance till here, and another compound travelled distance till here. So what you need to do to calculate RF value, you first need to find out A value. What is A? A is nothing but the distance travelled by the components from the baseline. From here, what distance is each component travelling? For that, from here to here, you mark the distance. This is taken as B. It is the distance travelled by the solvent from the baseline. And this distance is A. Then this distance is A. This distance is also A. For different components, you will have different RF values. So, if I choose for this compound, say if it is the distance travelled by the solvent is 6 centimeters and the distance travelled by this component is only 2 centimeter, then you will have one RF value. Then for the second component, this one, green one, say you had this value. Then for the third component, you have the same value. You have a different value. Each time B value, that is distance travelled by the solvent, it remains fixed. It remains same. What changes? Only the distance travelled by the component keeps on changing. So if you simplify them, each time you will get a different value, and based on the RF values, you can find out which compound has got more adsorbed and which component is very less adsorbed. So RF value gives us an indication about the relative adsorption of different components present in a mixture. Is that clear? Okay. So briefly, I shall revise this part, that is thin layer chromatography. Thin layer chromatography is a type of adsorption chromatography in which compounds present in a mixture are separated and identified based on the principle of adsorption. For this technique, a thin layer of stationary phase, either silica gel or alumina gel, is applied on a glass plate in the form of a slurry. At about 2 centimeters from the lower end of the chromo plate or TLC plate, a line is drawn. That line is called baseline. Then the plate is left for drying. On the other hand, the compounds which are to be separated are dissolved in a suitable solvent. We call that as a mobile phase. A drop of this solution is put or added onto the baseline, and this plate, TLC plate, is kept inside a glass jar containing the solvent or eluent or the mobile phase. Care should be taken that the solvent does not touch the baseline or the spot does not come in contact with the solvent. This plate is left undisturbed in the developing chamber for some time. After some time, the compounds get separated into different colored spots or patches. Now mark the solvent front line, and if the compounds are colored, then mark their places or their spots using a needle and let it for drying. After drying, scratch away the colored portions into separate beakers, redissolve them in a suitable solvent, decant them or filter them and subject it to distillation or evaporation, wherein you will get the desired organic compound. This is for colored compounds. If your compounds or components are colorless, then these three techniques can be used to identify the spots, either exposing the plate to UV light or else keeping the plate into a closed glass jar containing iodine crystals, due to which wherever spots are there, only those portions will become brown, or else by spraying suitable reagents on the plate, due to which only the spots will become colored or they will glow. One example for the third technique is identification of amino acids by spraying a reagent called ninhydrin. What are the advantages of thin layer chromatography? You can check the purity of an inorganic compound which has already been purified by some other method. This is one advantage. The second is even colorless compounds can be separated by this method. What is the disadvantage of thin layer chromatography? Large samples or organic compounds cannot be purified or separated by this method. In this thin layer chromatography comes another concept called RF value. What is RF value? RF value is nothing but the value which helps us to identify the relative adsorption of different components of mixture. It helps us to identify the relative adsorption of different components present in a mixture. RF value is given by an equation which relates the distance travelled by a component from the baseline to the distance travelled by the solvent from the baseline. Based on RF values, you can predict which compound can be adsorbed more and which compound is getting adsorbed less. So, this was about thin layer chromatography. Let us now study another chromatography. The second type is partition chromatography. It is partition chromatography. What is partition chromatography? Partition chromatography's best example is paper chromatography. In your second year, you have one experiment dedicated to paper chromatography in your syllabus. You will practice this chromatography in the labs. So, partition chromatography is a type of chromatography. It is now different from adsorption chromatography, but partition chromatography is a type of chromatography where the principles involved are partly partition and partly adsorption. This means 50% partition, 50% adsorption. It is also similar on the lines of adsorption chromatography, but the way of telling is different. In this technique, the compound which is to be separated gets partitioned between, stationary phase and mobile phase. To understand this, you need to understand what adsorption was. Adsorption means a surface phenomena where the compound remains only on the surface. It only gets adsorbed, it remains on the surface. So, whichever compound gets adsorbed strongly, it is separated late. It will travel less distance. This was the principle in adsorption. The same principle with a different way of telling comes in partition chromatography. I said partition chromatography is a principle in which a compound gets partitioned between stationary phase and mobile phase. This means whichever compound stays for a longer time with stationary phase, it gets separated late. Whichever compound has got less affinity for stationary phase, it will travel larger distances, therefore it will have more affinity towards mobile phase. So, it will move larger distances in presence of mobile phase and get separated easily. Based on that, we have paper chromatography as an example. What is paper chromatography? This looks similar to thin layer chromatography, but with what difference? Now, remember the difference is, in thin layer chromatography, we had a glass plate onto which stationary phase was coated. It was layered. What was that stationary phase? Either silica gel or alumina gel. Here in paper chromatography, we have specially designed paper, specially available paper. We call that paper as chromatography paper. It is made up of again cellulose only. It is again made up of cellulose fibers only, but it is a special kind of paper. That paper itself will act as a stationary phase. So, you need not remember any stationary phase for paper chromatography like silica gel, alumina gel, calcium carbonate, etc. No. Here a special paper made up of cellulose itself will act as stationary phase. So, now you imagine that this entire paper is our chromatography paper. It is nothing but chromatography paper. The procedure is same as that of thin layer chromatography. A paper is taken at about 2 cm from the lower end. A line is drawn, we call that line as baseline. Then you have 3 to 4 compounds which need to be separated. You dissolve them in a solvent. A drop of that solvent or a drop of that mixture is applied as a spot on the baseline. That is same. Now hang this paper or suspend this paper into a jar, glass jar called as developing chamber. Suspend or hang or drop this paper into a jar containing the mobile phase or the solvent required for elution. Then leave it undisturbed for some time. Upon leaving undisturbed for some time, the compounds present in the spot will migrate or will travel to different distances. Whichever compound spends more time with stationary phase, it remains nearer to the baseline. Whichever compound has got more affinity with mobile phase, so while the solvent is rising, it will travel to larger distances. Based on the distances traveled, based on the distance traveled by the solvent front, based on the distance traveled by the solvent front, what will you do? You will mark the areas. You will mark the areas covered by the compounds, and then only that portion is cut. In the thin layer chromatography, you scratched away the portions because it was made of glass. You scratched using a blade. This time you cannot scratch away because your paper absorbs them. So for that reason, you cut those colored part of the paper. After drying, you cut the colored part of the paper and then you add some solvent to that paper taken in a beaker. As a result, the solvent will take away all the compound and leave only paper. You throw away the paper, that colored solution is subjected to distillation. So this is paper chromatography. What is paper chromatography? It is a kind of partition chromatography. What is the principle involved? Partly partition and partly adsorption. How do I understand what is partition? A compound in paper chromatography gets separated based on the time it spends with the stationary phase or the mobile phase. Whichever compound spends more time with the stationary phase gets separated late. Whichever compound has got more affinity for mobile phase, it will travel with the mobile phase to larger distances, resulting in easier and faster separation. So for this reason, a special kind of paper made up of cellulose, we call that paper as chromatography paper, is taken. At about 2 cm from the lower end, a line is drawn, we call that line as baseline. On the other hand, the compounds which need to be separated, which are present in the form of a mixture, they are dissolved in less quantity of solvent, and a drop of that mixture is put on the baseline. Then this paper is hanged or suspended in a jar containing the mobile phase. Leave this apparatus undisturbed for some time, and then you will notice that the compounds present in the mixtures have rows to different heights based on their partitioning between stationary phases and mobile phases. Once when all the components get separated into different colored spots, then you take out the paper, let it for drying, let it be kept for drying, and after drying, if the colored portions are clearly visible, then you cut only those colored portions into different beakers. Different colored portions into different beakers, add solvent to it, stir well so that the compound adsorbed on the paper comes along with the mobile phase. Throw away the paper or filter the solution and then subject it to distillation to recollect the solvent as well as the compound. Now what if the compounds are colorless? On the paper you can only see the solvent front but not the different colored spots because your compounds are colorless. Then what you will do? You will again use the same 3 methods that you used in thin layer chromatography. One, you expose the plate to UV light, or else keep the plate, you expose the chromatography paper to UV light, or else you will suspend that paper into an iodine chamber, due to which spots only will become brown, the paper will not. Or else you spray a suitable reagent on the chromatography paper which helps in easy identification of the colorless components. So by this we have studied how many purification techniques? 1. Sublimation, 2. Crystallization, 3 distillation, 4 solvent extraction and 5 chromatography. So this completes 5 different types of purification techniques. In our next classes or in our next sessions, we shall see how different elements present in an organic compound can be detected, and then we shall go for their estimation. So keep watching the classes. Thank you.